Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 20th July 2020. And in this session we are going to introduce new chapter called Biomolecules, and this is all the while important because it is there in your chemistry also. And the effort is that if you understand here, I think it would be very easy in your chemistry also, and in general in your life. It is always better to know what is inside us. And what is inside us? Lot of biomolecules. And what are biomolecules? These are chemicals basically. These are chemical molecules present in all of us living organisms. And they are responsible for bios, life. Because of their presence, we are having life. So the total biomolecules that are present inside us constitute a pool, just like a swimming pool, like that, a cellular pool. And basically these are derivatives of carbon. Carbon is the main compound in that. Hence the chemistry of living organisms is organized around carbon. And carbon is most versatile and predominant element in our life. And look at the percentage that is there in our living matter. Carbon 18.5, and of course, and that is the reason why all the cells will be having their constitution with carbon. So hydrogen you got in living matter, we are focusing on living matter 0.5, carbon of course 18.5, oxygen 65, nitrogen 3.3, sulfur 0.3, sodium 0.2, and calcium 1.5, magnesium 0. And silicon. So carbon is having a crucial role in that. And in biomolecules we can divide them into two, organic and inorganic. As you know, organic refers to carbon. Carbon will be playing a crucial role in that. So we have got under organic biomolecules carbohydrates, lipids, amino acids, proteins, enzymes, nucleotides, nucleic acids, and vitamins. Under inorganic you got minerals, gases, and water. So this is how we classify. And further, based on their weight also we classify biomolecules into two types, micromolecules and macromolecules. So micromolecules, we measure their weight in terms of Dalton. Remember that great scientist, Dalton's atomic theory. Yes. So their low molecular weight, 18 to 800 Daltons, and they are found in acid soluble pool. So what are those? Minerals, gases, water, sugars, amino acids, and nucleotides. We'll talk about this nucleotides quickly. And then macromolecules, large sized, high molecular weight, above 10,000 Daltons, found in acid soluble pool. They are carbohydrates, lipids, proteins, and nucleic acids. And the major complex biomolecules of cells. Proteins, of course, the building block is amino acid. That means one amino acid plus one amino acid together will make a protein. And major function, all of you know, without proteins, they are building blocks of the cell. So without proteins, there is no construction of cell at all. So major function of protein is cell construction. So basic structure and function of the cell is decided by what type of protein is present in the cell. Next, DNA. Expansion of that is deoxyribonucleic acid, and this is made up of deoxyribonucleotides, and they have what is our secret, that is genes. So DNA has got our genes, which are hereditary information centers. And then RNA, ribonucleic acid. This is also called ribonucleotides, and it helps directly in protein synthesis. Hence, without RNA, there is no protein synthesis. Without proteins, no amino acids. Without amino acids, no cell. That is the logic. And then polysaccharide. Poly means many. Saccharide means sugar. Many sugars. And they are made up of, they are called monomers. And many monosaccharides join together and form a polymer, and the polymer is polysaccharide. Like for example, starch is a polymer, and starch is made up of many monosaccharides, and they are responsible for energy, storage of energy. And lipids, the other name for lipids is fats, and fats are made up of fatty acids and glycerol, and they are mainly responsible for energy and also gives us warmth. And lipids play a very crucial role in maintaining the temperature, body temperature. And then we come to all important nucleotides. What is a nucleotide? A nucleotide is a unit that makes DNA or RNA. And just now I said DNA is deoxyribonucleic acid. RNA is ribose nucleic acid. What is the difference? Deoxy. In a sugar called ribulose or ribose, one oxygen is removed. That becomes deoxy. D means minus of oxygen. So DNA. RNA, there is a ribulose sugar, oxygen is there in that. So whether it is DNA or RNA, both are made up of small units. Those units are called nucleotides. And what is a nucleotide made up of? First, you must remember there is a nitrogen base. Look at the nitrogen molecules present there. Nitrogen base. And then one sugar molecule. This is the sugar we are talking of. That is ribulose sugar. It's a pentose. Penta means five sides are there, you can see. So there is a pentose sugar. And then to that pentose sugar, on one side, phosphate group is attached. That is phosphoric acid. Okay. And the other side, nitrogen base is attached. So a sugar having a pentose and nitrogen base is a nucleotide. And such nucleotides, we can consider this as a monomer. Many monomers like this will join together, make a polymer, and that polymer is either DNA or RNA. And now look at this, the deoxyribose and ribulose. Just now I said no, the difference is O. And focus on this blue ink and pink one. Here on second carbon you got only hydrogen, where in deoxyribose. Whereas in ribose sugar, in second carbon you can see the hydroxyl group, OH group. So between deoxyribose and ribose, what is missing? One oxygen atom is missing. That's why we are saying it as a deoxy, minus of oxygen, where the minus is taking place on second carbon. I am sure in your chemistry you have learned the way you number the carbon atoms, like 1, 2, 3, 4, 5. And that's why we say it's a pentose one, penta, 5. And then nitrogen bases. Nitrogen bases, there are two, purines and pyrimidines. Now please look at the diagram. Purines are double ringed structure. Two rings are there, 1 and 2. Whereas pyrimidine is only one ring, single ring structure. So purines are two types, adenine and guanine. Adenine is given symbol A. Guanine is given symbol G. A, G, you must remember. So how to remember this? PAG, PAG. Purine or adenine and guanine. PAG, PAG. And pyrimidine, it has got 3. Cytosine, uracil and thymine. So here you can say pyrimidine, PY you can say. PY you can say, and you can remember it as PIC UT, PIC PY, because there also purine is there top. Here you can remember it as PY, pyrimidine, and cytosine, first letter C, PIC, PYC, and UT, Union Territory, uracil and thymine. And cytosine and thymine, now you must differentiate here that uracil is a pyrimidine that is present only in RNA, and in DNA, in its place, thymine will be present. So thymine will be in DNA and uracil will be present in RNA. And I want you to draw this as you are seeing this picture in your classwork. Please draw the diagrams, practice it at least 3 to 4 times. Then only you will be familiar with the numbering of carbon. And then nucleotides. Just now we said based on the attachment of sugar molecule. One side you have got nitrogen base attached, nitrogenous base attached. On the other side, phosphoric acid is attached. So based on what structure it is attached, for example, adenine is attached. Then we say the nucleotide is deoxyadenylate or deoxyadenosine. In nucleoside, when we say nucleotide and nucleoside, deoxyadenosine. And similarly, when we go for guanine is the nitrogen base. Sugar is there. One side guanine is attached, that is first carbon, and fourth carbon the phosphoric acid is attached. Then we say deoxyguanosine. If deoxyadenosine with adenine present, guanine if it is present, then it is automatically called as deoxyguanosine. Similarly, deoxythymidine. Similarly, deoxycytidine, cytosine is present. And in deoxyribonucleotides, you can see those. And what about in RNA, ribonucleotides? In ribonucleotides also we say adenosine, that is adenolite, okay, and guanolite, that is guanosine 5-monophosphate, GMP we say, and uracil is present, that's why it is uridylate, and cytosine cytidylate. And this is how the DNA having varieties of nucleotides will be present. And we can say we must be thankful to two scientists, Professor Watson and Crick, and they got Nobel Prize for discovering the structure of DNA. This structure is called double helix. Helix means nichanige, double helix. If you twist the ladder, this is how it becomes, and DNA correspondingly named as double helix. So you got two vertical threads, and those vertical threads are made up of sp. Remember sp, sugar and phosphate, and the two vertical strands are interlinked with horizontal steps. Those horizontal steps are nitrogen bases. One purine is attached to one pyrimidine and vice versa, and there is a base pair rule. Adenine always pairs with thymine, AT, adenine and thymine, and guanine always pairs with cytosine and vice versa, GC. So you got AT or TA, GC or CG. This is how the base pairing takes place. And you can see here the structure, internal structure. You can see the weak hydrogen bonds making the attachment of nitrogen base, like G for guanine, C for cytosine. You can see triple bond between G and C. There is a triple hydrogen bond, and between A and T or T and A, you can see a double hydrogen bond. So this is called complementary base pairing. All this structure, you can imagine, have been discovered by Professor Watson and Crick, and they deservingly got a Nobel Prize for this. And once we open up this, that's called replication of DNA, we understand the secrets of the genes, what the secrets the genes are containing. So DNA is helping us to understand that. And this is how the double helix model and the original DNA looks like. You can see here the major groove, minor groove, and there is something called direction, 3' to 5' and 5' to 3'. That is position of the carbon atoms. So don't involve yourself much in this. We'll be studying in 12th class in detail about the structure. And coming to RNA, RNA is of three types. One is, basically you must remember, DNA has got two threads, so it's a double helix, okay? RNA has got a single thread, only one ribbon, and depending upon the functions that RNA performs, it is of three types. mRNA, messenger RNA, means it collects messages and pass on the messages from one area to another area, so it is called mRNA. Second, rRNA, ribosomal RNA. This RNA will be having ribosomes on it. That's why it is called ribosomal RNA. And then tRNA, that is transport RNA or transfer RNA. And look at the speciality of this tRNA. It is like a clove, lavanga, clove shaped structure. And all these three will help in what we call protein synthesis. And particularly greater role is played by mRNA and tRNA. And the tRNA has got what is called amino acid arm and anticodon arm. Now you can see the anticodons and codons, Uebel position, all this we will discuss when you come to 12th class. At present, just you remember that RNA is of three types: mRNA,